Hi, my name is Tim Liu. I'm a professor at MIT. I'm here to tell you today about synthetic biology, an emerging engineering discipline. One of the challenges in defining the term synthetic biology is that it's a term that's used to mean different things to different people. And I've listed several of the common definitions that people use for this particular term here on the slide. Regardless of how you exactly define synthetic biology, I think this is a community that's unified by the desire to engineer biological systems for new functions, whether we're doing this from the ground up, from minimal systems, or from the top down, taking an existing system, trying to retrofit it to do new things. This is a robust and vibrant community that is growing. I come from the community at MIT. We have the Synthetic Biology Center there, where we're actively engaged in various aspects of engineering biological systems. Globally, there's a growing community of labs interested in the field of synthetic biology, and this is well evidenced by the iGEM competition, which has grown from a very small community to a very large global community that comes together every year to compete in terms of genetically engineered machines. From an engineer's perspective, one of the exciting things about the field of synthetic biology is that we're moving from an era where we were simply cutting and pasting DNA from one place to another to a point where we're able to specifically program the A's, T's, G's, and C's that go into biological software. And in some sense, this is really a technologically driven improvement. So here is a graph showing comparison of Moore's law, shown in blue, which is named after one of the founders of Intel, Gordon Moore. He basically predicted that the semiconductor industry would be able to double the number of transistors on a given chip every 100 years. And this exponential scaling in computing power has led to tremendous advancements in our ability to program silicon-based systems. Now, if you look at the other two curves on this graph, you can see that our capabilities for DNA sequencing and DNA synthesis are also increasing at an exponential rate. And therefore, our raw capability to program and read DNA is increasing every year. That's really giving us the raw capability to modify biological systems to do new functions. However, there are multiple challenges that are associated with engineering biological systems that are very different from conventional ways of engineering electrical circuits. So for example, in electrical circuits, you can take components and explicitly wire them together to achieve new functions. Whereas in biological systems, parts that we develop are bouncing together in the cell and really are wired based on chemical specificity rather than physical wiring. And this poses a challenge when you're trying to scale and build complex biological circuits. As a result of this implication, in electrical circuits, you can actually duplicate the same identical device, the same transistor, over and over on a chip, and wire these identical devices together to achieve some more complex function. Whereas because biology requires specific chemical wiring between components, you need to develop unique devices to implement every additional level of complexity you're trying to achieve. Electrical circuits have had many years to try to develop formal languages and designs that allow you to compose more complex functions, whereas the field of designing biological circuits is in its relative infancy, where there are still new concepts and new circuit designs being developed every day, and therefore no kind of dominant formal language being present to describe how to program biological systems. The dream of the field is really to start from the bottom up, to be able to program the DNA code, and to start developing devices and circuits and modules that have additional layers of complexity, with the ultimate dream of to build very complex systems and networks, something like a whole organism that carries out a specific function. We're still quite away from the ultimate dream of kind of engineering a very complex system, but we have made a lot of progress in terms of building devices and circuits and starting to layer these together to higher order modules, which I'll describe in my talk today. The idea I'd like to communicate to you today is that biological circuits are at the core of synthetic biology because they enable complex sense and respond behaviors from cells. So cells can be thought of as little mini computers in that they sense all sorts of information. They process that information so they can perform computations, but they can also store the output of the computations in memory. And then the cells can respond by carrying out new functions, whether it's deciding to live or die or divide or not, or produce a certain molecule. So at the core of this, we need the ability to program biological systems to compute information and to store this information in memory, because this will enable us to build very generalizable frameworks for sense and respond behaviors. The other thing I'd like to talk about towards the end of the talk is really how far we are in this field from being able to rationally program biological systems that carry out specific functions. So we'd like to really adopt the engineer's framework where we can go from concept to design, model, simulation, where we kind of develop our designs in silico. Then we experimentally construct, probe, and measure these systems and iteratively improve our systems. And I'll talk about some of the design challenges that are still remaining in the field that make it challenging to implement this entire pipeline. The other interesting thing about biology is that unlike silicon-based systems or other engineering disciplines, we have the added capability of evolving biological systems. And that could present actually interesting ways of trying to design systems that carry out new functions, but also poses a challenge when you're trying to design a biological system that carries out a function, and we want the cells to continue to develop that function or display that function without evolving away from it due to the other evolutionary pressures. And so this is a unique challenge, an opportunity for biology, this idea that you can incorporate evolution into the design process. So as I mentioned, there are many flavors of synthetic biology, and even in the field of circuit design, there are many reasons why people are trying to design biological systems. For example, people are interested in solely the idea of computation. Can you design circuits that can allow you to compute functions inside of living cells? Other folks are interested in this idea of therapeutics. So if you could design cells that carry out a specific function, could you use that to program new cell and gene therapies that are responsive to their environment? Other folks are interested in producing new molecules. So if you could design circuits that can sense metabolites or sense intracellular state, could you optimize the expression of new heterologous materials that cells are making, such as chemicals or other physical materials? And finally, there are folks who are interested in this idea of using genetic engineering or synthetic biology to build circuits to understand biology at a deeper level, with the idea that if I cannot build a system, then I cannot fully understand it. And therefore, people are interested in building genetic circuits as a way of understanding basic principles of how biology functions. So before I get into the deep circuit engineering